Thank you, Professor Charlie and Redmark, for a kind introduction. So it is my great honor and pleasure to be here together with Fred Ramsdale and Alexander Rudinsky to give a lecture today. So the title of my talk is Control of Immune Responses by Regulatory T Cells. So there is one important concept in immunology, that is immunological self-tolerance. So the immune system, it reacts with non-self, such as bacteria and viruses, and protect our body. On the other hand, it shouldn't react with self or destroy self-constituent. If it destroys our body, it causes, just as Professor Redmark mentioned, it may cause autoimmune disease. And sometimes the immune system reacts with the non-toxic, just environmental substance like a pollen and cause allergy. And chronic viral infection, microviral infection, for example, in the intestine, we have the vast amount of intestinal bacteria. But our immune system normally doesn't react with commensal bacteria in the intestine. So, and in medicine, we hope that immune system it will react with the cancer cells which have arisen from self tissue. Or ideal of organ transplantation is to make the immune system look at other body's organ as if it is self. So, and also fetal-maternal tolerance, it is still an enigma why the mothers can harbor a baby, he or she is not herself, and why she doesn't reject babies, the fetus. So it's called fetal-maternal tolerance. So we have been working on this concept, immunological self-tolerance, for many years. And let me first start with autoimmune disease. So as Professor Redmark already mentioned, there are many autoimmune diseases, one called organ-specific, such as multiple sclerosis or thyroiditis or gastritis or type 1 diabetes, and systemic ones, the typically rheumatoid arthritis, which Holger Crafoord suffered from. So cause and the mechanism of these autoimmune diseases still is not clear. And immunology, well, from the 1950s, the modern immunology has proposed several mechanisms. The main mechanisms are three. One is that the developing T cells or B cells, when they react with self-recognized self-antigen, they put to death by apoptosis. It is called deletion, clonal deletion. A second mechanism is they may recognize self-antigen, but it's not enough to activate them. They need another the stimulation. It is called co-stimulation. Just recognizing antigen, they become inactivated. In immunology term, it is called anergy. And third mechanism is our immune system, it harbors hazardous, dangerous self-reactive T cells. But we also harbor the kind of immune suppressive T cells. Now we call them regulatory T cells. So if you remove the regulatory T cells, no suppression, so these self-reactive T cells get activated and cause autoimmunity. So deletion, inactivation, or suppression. So which mechanism can explain real autoimmune disease? In other words, by manipulating which mechanism we can produce the autoimmune diseases which we see in humans. So one important feature of human autoimmune disease is overacting. So if you see the patient with type 1 diabetes, he or she may have also a thyroiditis or gastritis or adrenalitis. So this kind of overlapping is very common. And it is also common in animals. For example, this is NOD mice which spontaneously develop type 1 diabetes. But when they become aged, they develop thyroiditis. And the BB rats, this is also a spontaneous model of type 1 diabetes. But they do also develop thyroiditis and gastritis. So these findings altogether, the question is autoimmune disease due to anomaly of the immune system or target self-antigen? And from that point, point of view, there are historically two interesting experiments conducted many years ago. So one is the neonatal thymectomy, just normal mice, and then you remove the thymus, producing T cells around day 3 after birth, and then 2 months later, you'll see some autoimmune diseases, autoimmune overacting. This is autoimmune disease in ovaries, or gastritis, stomach, or thyroiditis, a thyroid gland. And this was found by 2 Japanese pathologists, Nishizuka and Sakakura, in 1969. And another interesting model is using rats. So normal rats, and you remove the thymus around 6 weeks of age. And every 2 weeks, if you irradiate 200 Röntgen, 2 gray, it's a sublethal dose, just 4 times. And then these normal rats, they developed autoimmune thyroiditis or type 1 diabetes. So this means that just manipulating the immune system, you can induce various autoimmune diseases, immunopathologically similar to the human counterpart. So this is neonatal thymectomy, valve C, A, and C57 black 6. These are different strains of mice, and they're all normal mice. There's no autoimmunity. But if you remove the thymus within 24 hours after birth and day 0, you don't see the autoimmunity. But on day 3, now you see a 30% gastritis, 30% ovaritis, but day 7, nothing happened. And day 3 thymectomy of this strain, now 90% developed ovaritis and with some other autoimmunity. But on this strain, another strain, this strain is resistant to autoimmune induction by neonatal thymectomy. So critical DNA of thymectomy is important, and the genetic background is also important. And how this happens, what's the mechanism? And one important finding is that after a thymectomy, or before thymectomy, or after thymectomy, if you inoculate normal T cells from normal adult mice, now you can prevent disease development. For example, here, just neonatal thymectomy, the 80% or 90% develop ovaritis, autoimmune disease in ovaries. But if you inoculate 5 million T cells before thymectomy, or 1 week after, or 2 weeks after, completely you can prevent disease. So this means that in normal T cells, well, normal T cells may contain a sort of immune suppressive lymphocytes, T cells. So this is how neonatal thymectomy causes autoimmunity. Normal thymus produces T cells from gestational days on. Day 0, the mice are born. And day 3, if you remove the thymus from on day 3, now you can block the migration of the newly produced T cells into the periphery. So why autoimmune disease happened? So perhaps the reason is that those T cells that are produced by the thymus may contain the potentially dangerous the autoimmune T cells. And also, around day 3, thymus may produce the kind of the immunosuppressive T cell, regulatory T cells. So if you remove the thymus before the day of thymectomy, some T cells already in the periphery, they get activated and cause autoimmunity. So if you inoculate normal T cells, you can suppress these autoimmune T cells. To demonstrate this hypothesis is correct, the most critical experiment is to separate regulatory T cells and autoimmune T cells. And then not by thymectomy, just by directly, if you remove this population, you can ask whether the autoimmune disease happened or not. So this is the experiment we did, normal mice. This is spleen. Spleen is packed with the lymphocyte, T cells and B cells. And then we attempted to remove a kind of immune suppressive cells by cell surface markers. So various cell surface markers, and then we removed, T cells expressing this marker, remove, and then remaining cells into T cell deficient mice. So nude means they have no thymus. So B cells are okay, antigen presenting cells are okay, but no T cells. So if you transfer after removing a sort of immune suppressive cells, these mice may spontaneously develop various autoimmune diseases. That was indeed the case. Many years ago, we first using this marker, CD5, and then 5 years later, a group in Oxford, they used another marker. And then we wondered by removing the smaller number of cells, and then we can induce autoimmunity in the wider spectrum of organs with more severe form. That is more specific marker for this population. And then we came up with CD25. So CD25 is the IL-2 receptor alpha chain, and CD4 T cells, 10% expressing the CD25, but then less than 1% in CD8. And really, they are CD5 high, and CD40 RC low. So it's a good candidate. And then indeed, if we remove this 10% of population and then transfer the remaining cells into nude mice, they did develop just similar to human autoimmune diseases, thyroiditis, gastritis, and type 1 diabetes, and so on, and also inflammatory bowel disease, which is the abnormal reaction to commensal bacteria in the intestine. So the stomach destroyed. You see many blue color cells. They are lymphocytes coming into to infiltrating into the gastric mucosa and destroying it. And you can demonstrate autoantibodies against gastric parietal cells, as shown here. And this is thyroid gland. So here thyroid hormones are stored, but here lymphocytes infiltrate and then destroying the thyroid cells. This is Langerhans islet. So you see the infiltration of the mononuclear cells, lymphocytes, into Langerhans cells. And other Langerhans cells all islets are already destroyed. And actually this mouse developed type 1 diabetes. So just removing a small number of population, the CD25 positive cells, you can induce various autoimmune diseases. So these findings altogether suggest to us that what is important is a balance between suppressive T cells, now regulatory T cells, and autoimmune T cells. So this balance is impaired, for example, by reducing the number of Treg or impaired their function. It's enough to cause autoimmunity because self-antigens are always in our body. So just removing the suppression is enough to cause autoimmunity. So the helper cells help B cells to secrete autoantibodies or activate killer cells, cytotoxic lymphocyte, and or recruiting macrophage or other inflammatory cells and destroy self-tissue. So we have shown that whatever it is, environmental insult or genetic anomalies, if they affect the balance, it can cause autoimmunity. And so certain viruses or certain chemical or just I mentioned to you, ionizing radiation, all can relatively more affect the regulatory T cells and then cause autoimmunity. Then you may ask then, it's really true in humans. And then we came up with the FOXP3. So around year 2000, Fred Ramsdell and his group showed that the cloned the FOXP3 gene as responsible for scurfy mouse, very severe autoimmunity, and they named it the scurfin or FOXP3. And immediately it was found that this is responsible for human disease called IPEX syndrome. So IPEX means that immune dysregulation, polyendocrinopathy, so the various autoimmune diseases in endocrine organs like type 1 diabetes, and enteropathy, inflammatory bowel disease, and disease genes on X chromosome. So more than 80% of patients develop type 1 diabetes within 3 years after birth, more than 70% thyroiditis and other autoimmunity. Almost 100% inflammatory bowel disease, and very severe diarrhea, and allergy. So these patients cannot take food orally because soon they develop a food allergy. So this is very similar to what we saw after Treg depletion. So we wondered the FOXP3 is something to do with regulatory T cell development or function. So this is our antibody against FOXP3. So the normal CD4 T cell, 10% express some FOXP3, but the scurfy mice with FOXP3 mutation, no such cells. And if you stain for FOXP3 and CD25, so here in the thymus and spleen, the majority of FOXP3 positive cells, they are CD25 in the spleen and also thymus. And so in the previous years, we removed the CD25 positive cells. That means we remove the majority of FOXP3 positive cells, so we could induce autoimmunity. So now FOXP3 is the best molecular marker in the nucleus, and then CD25 is the best marker on the cell surface to define regulatory T cells in rodents and also in humans. To summarize this part of my talk, first we showed with mice that if you remove a certain population of CD4 T cells here, and if they transfer the remaining cells into T cell deficient mice or TB cell both deficient mice, they develop the various autoimmune diseases, IBD, and the inflammatory bowel disease, and allergy type hyperreactivity. Just similar process is going on in humans. So IPEX patients, the disease gene on X chromosome. So mother is the normal X chromosome and the abnormal X chromosome. So, but it's a kind of mosaic. So normal one suppress the immune responses, so no disease. But then patient, usually there are male kids because HY, the XY, so X chromosomes are abnormal, then the Tregs are either in function or development, they are abnormal. So they cannot suppress the self-reactive lymphocytes or the lymphocytes causing allergy. So what's going on in humans and in mice, they fit very well. So this is the view of the immune system. So we do harbor the potentially dangerous self-reactive T cells, normal individuals, but they are controlled not to be activated. And if you remove Treg, the immune responses against self, now it triggered. So you see common autoimmune disease like diabetes and thyroiditis, and also the immune responses against bacteria or the viruses, for example, against intestinal bacteria. So you have the immunopathological disease like inflammatory bowel disease, and also against environmental substance causing allergy. So this means that the border between self and non-self is not fixed. It can be movable and not so fixed. So this means that by targeting Treg, either increasing in number or function, or decreasing their number or function, you can control the immune responses. So it is the one thing. It is which is interesting is that in developed countries like Scandinavian countries, so the infectious diseases, the incidence become lower and lower. So in these 50 years, from 1950 to 2000, this work was reported by Jean-François Bach and French immunologists. It is a well-cited the article published by his group. So infectious diseases decreasing, but on the other hand, autoimmune disease and the inflammatory bowel disease, such as Crohn's disease, and the allergy, asthma, all are increasing in developed countries. So this means that they are very similar to what we see after Treg depletion. So perhaps Tregs are less trained in hygienic societies. So like muscle, if you don't train your muscle or moving muscle, it becomes weakened. Similarly, the lymphocytes must be trained to have proper immune responses. So not too strong, not too weak, but proper level of immune responses. And that is important for controlling the hazardous lymphocytes in our body, like the T cells causing autoimmunity or causing allergy or inflammatory bowel disease. Now, let me move on to a little bit about immunology, more detailed immunology. So here, FOXP3 positive Treg, they are produced by the normal thymus. So highly self-reactive T cells are deleted. So deletion is real. But then also thymus produces regulatory T cells to control, suppress the anomalous immune responses. So the question is, how Treg suppress immune responses? And so FOXP3, this is important for Treg function. And I believe most important function is if you express FOXP3 in normal T cells, you can convert, change the normal T cells to regulatory T cells. So this is retroviral vector, incorporating FOXP3 gene here. And then if you infect the normal T cells, now the 40% become GFP positive, green fluorescence. And by removing Treg, then if you transfer them into normal mice, T cell deficient mice, you have colitis, inflammatory bowel disease, and autoimmune gastritis. But if you co-transfer with FOXP3 expressing transduced conventional normal T cells, now you can prevent or suppress the disease development. Just control empty vector transduced cells, as they cannot suppress. So this means that FOXP3 controls the downstream suppressive machinery. So FOXP3 mutation causes a very severe autoimmune disease, and the question is, if you disrupt the downstream, which gene, if we disrupt a similar autoimmune disease, would happen or not? So not so many genes. FOXP3 itself controls the hundreds of genes, and FOXP3 bind to 1,000 of the genomic site in normal individuals. But the knockout mice, not so many knockout mice develop a severe autoimmunity. So only 4. One is IL-2 knockout, and second CD25 IL-2 receptor alpha chain knockout, and CD122 IL-2 receptor beta chain knockout, and another one is CTLA4. So FOXP3 they activate CD25 and CTLA4, but then they replace an IL-2 or interferon gamma gene. So let me talk about IL-2 receptor and CTLA4. So the IL-2 receptor, which is composed of the alpha beta gamma chain, and then we see the high affinity IL-2 receptor. So a cardinal feature of Treg is they are constitutively expressing the high affinity IL-2 receptor. So usually lymphocytes when they are antigen exposed to antigen and become activated and differentiate, so then they express a high affinity IL-2 receptor. But Tregs are already expressing a high affinity IL-2 receptor in the thymus. So here, the CD25 means IL-2 receptor alpha chain positive and beta chain positive and common gamma chain positive. But CD25 negative cells, alpha chain negative, they are not expressing beta chain. If you go to the thymus, alpha chain positive cells are expressing beta chain and common gamma chain. So this means that already in the thymus, they are expressing the high affinity IL-2 receptor. And if you neutralize IL-2 in the body, the Treg cannot survive. So just one injection of a neutralizing anti-IL-2 antibody, now the Treg in the spleen, now they decreased in number, and also in the thymus. So this means that by neutralizing IL-2, now we can induce autoimmunity. So just in normal mice, if you inoculate anti-IL-2 just twice after birth, it's enough to cause the autoimmune gastritis. And this is the NOD mice, which are prone to develop thyroiditis or the type 1 diabetes. Now you see very severe type 1 diabetes and thyroiditis or thyroadenitis or lacrimal gland, the adenitis, autoimmune disease in these the salivary gland and lacrimal gland. So just neutralizing cytokine, one cytokine, IL-2, that's enough to cause autoimmunity. So because Treg need IL-2 for survival, by removing IL-2, Treg cannot survive, and you see Treg depletion, and you have autoimmunity. How about CTLA-4? Perhaps you may be aware of it. The anti-CTLA-4 antibody is now clinically used to trigger, to augment the tumor immunity, cancer immunity. So our understanding is that the T cells, when they recognize antigen, now it's called signal 1, and then, but they need signal 2 co-stimulation for their activation. Once they are activated, they express on CTLA-4. It's a homolog of CD28 and interacting with CD86, but now they put negative signal, so the immune response can be subsided. But interestingly, regulatory T cells are always expressing CTLA-4, very high level. So the question is, if we block here, perhaps what happened to Treg, whether they cannot suppress immune responses, and we may have autoimmunity or not. So it was indeed the case. So normal biopsy mice, just day 10 and 20, and then we inoculated anti-CTLA-4 twice, and 3 months later, very severe autoimmune gastritis developed. So just like a Treg depletion, anti-CTLA-4 blockade, it's enough to induce autoimmunity. And so here, the FOXP3 positive cells, they're expressing CTLA-4, and we made a CTLA-4 conditional knockout mice. The FOXP3 positive cells, Tregs here, but they don't express CTLA-4. And what happened is that they died of very severe autoimmunity, for example, autoimmune disease in the heart. So this is a ventricle. These blue parts are the infiltrating lymphocyte. This is the atrium. All cardiac myocytes are destroyed. And in the circulation, a high concentration of IgE, meaning that they are prone to have allergy. And importantly, they have a higher the cancer immunity. So we transferred the T cells into nude mice and then inoculated cancer cells here. So red one, conditional knockout mice without CTLA-4 expression. The immune response is the enhanced. So the tumor first become bigger and then rejected. But the control mice all died of tumor progression. So just like by Treg depletion, by non-expression of CTLA-4 by Treg, it's it really causes a similar autoimmunity or tumor immunity and allergy as we see in FOXP3 mutation or in human IPEX syndrome. So now in humans, the CTLA-4 antibody is in now clinical use. So this is the first report in 2003. They reported that the cancer regressed, but at the same time, autoimmunity developed. So here, CR means complete response, but at the same time, this patient developed autoimmune disease in hypophysis, and the complete response and dermatitis or vitiligo skin disease and the colitis, inflammatory bowel disease. So just similar to what we see in mice, similar thing going on in humans after anti-CTLA-4 antibody treatment. So how this happens? So really CTLA-4 control the co-stimulatory molecule expression by antigen-presenting cells. So here in vitro, this is dendritic cells expressing a co-stimulatory molecule CD86, and if you co-culture with normal T cells, the T cells are activated, and further CD86 is upregulated. But in the presence of Treg, red arrow, there is no such upregulation of CD86 or CD80. And just similarly, in the presence of Treg and responder cells altogether, the Treg suppress the upregulation of these co-stimulatory molecules, CD80 and 86. So this is a current understanding. FOXP3 transcriptional complex, it controls many genes, but an important function is repression of IL-2. So, but upregulation of IL-2 receptor. So they don't produce IL-2. So they are highly dependent on the exogenous IL-2. So if you neutralize IL-2 or IL-2 knockout mice or IL-2 receptor knockout mice, Treg cannot survive, and you'll see autoimmunity. And second important molecule is CTLA-4. And if CTLA-4 is expressed constitutively, it interacts with a co-stimulatory molecule, CD86, and down-regulates the expression. And IL-10, further Treg differentiates, they produce IL-10, which also controls the APC. So as a result, the IL-2 deprivation and deprivation of co-stimulation, the responder cells cannot be activated. Now the question is, what would happen to these responder T cells under control by Treg? So let me go through quickly. Now time is running up. So this is human peripheral blood, CD8 T cells and the Treg. So the antigen-presenting cells together, and then without or with Treg, 7 to 10 days, and we assess the CD8 positive cells, whether they are proliferation, cytokine production, or T-cell affinity. The antigen we use, the melan A, this is self-antigen, melanocyte differentiation antigen. And then this is the target antigen of vitiligo vulgaris, an autoimmune disease of the skin. And this is also a tumor-associated antigen of malignant melanoma. So with Treg, as I told you, that the upregulation of CD86 red line is inhibited. And in the presence without Treg, now these melan A specific T cells, self-antigen specific T cells, now they can expand. But if you add Treg in gradually, now the response can be suppressed. 1-to-1 ratio, almost no such cells. So really, the suppression is operating in this system. And more importantly, if you label the cells with CFAC, so this to look at the dividing cells. So the T cell divide 1, 2, 3, 4, 5. This is the CFAC labeling. So without Treg, the melan A specific T cells expanded many times in cell division and form this peak. But in the presence of Treg, they only divided once and accumulated and forming this peak. So if you compare A and B, so by if you stimulate them, then A can proliferate, but B cannot proliferate, no cell division. And A can produce various cytokines, interferon gamma, TNF alpha, IL-2, but B, they cannot. So this means that this is the definition of energy. So proliferation aborted and cytokine hypoproducing. So this means that Treg put the responder T cells into anergic state when the Treg are present. And if you compare TCR affinity, so the melan A means tetramer. This means this melan A specific T cells are stained here. It's higher stained. This is a higher affinity. If it's lower, it's a lower affinity. So if you compare A and B, A is higher in affinity. And then what we found is that in the presence of Treg, some T cells become anergic, but a higher affinity one, they disappeared. So this means that in the presence of Treg, some the low affinity one become anergic, but the high affinity one, they are put to death by apoptosis. So this is what we see in these experiments. So Treg controlled APC antigen-presenting expression of a co-stimulatory molecule. So depending on TCR affinity, if it's high, they are deleted. If it's intermediate, become anergic, or low, they are still dormant. If you remove Treg, the APC is highly activated. So self-reactive T cells can be activated and cause autoimmune diseases. So now we can define anergic cells by cell surface antigen. So in Treg history, we first defined address the question how we can define Treg objectively. So similarly, anergic cells, how we can define them. So we did various assays, and what we found is that these anergic cells are naive phenotype, meaning that the CCR7, CD40RA, CCR7, the chemokine receptor, if they express, this means a kind of non-activated, naive T cells, but also they are expressing CTLA4. So by using CCR7 and CTLA4, now we can define anergic cells, so activated T cells or naive T cells, very differentially. And now the patients. So healthy donor, self-reactive melan A specific T cells, small number, 0.03%. But a vitiligo patient, so the skin become white because the melanocytes are destroyed. This is autoimmune disease. Now they expanded. But a phenotype, in normal individuals, we call them a naive phenotype, CCR7, CD40RA high. But a vitiligo patient, they are activated. And importantly, these in healthy donor, they are in anergic state because 0.03%, and you staying for CCR7, CTLA4, the majority of them are CTLA4 positive, and some of them are CTLA4 negative. And the CTLA4 positive one, they don't proliferate, but the negative one, they can proliferate. The positive one, they don't produce cytokine, but the negative one can produce. So in our body, Tregs are present. Tregs are suppressing potentially dangerous lymphocyte, and some of them become put to anergic state. So they will eventually die. But also naive T cells here. If you remove Treg from this population, autoimmune disease, the causing lymphocyte proliferate, or if you remove Treg, now the T cells react with cancer cells, become activated. So if you vaccinize with cancer antigen, these naive population, they can be activated to cause, to have a good tumor immunity. So here, this is summary. So the Treg controlled APC, and by APC, the responder cells are controlled. Depending on TCR affinity, they may become apoptosis or anergy or dormant. And what is important is Treg-mediated suppression for limited period is able to establish long-term immunological tolerance by determining cell fate of responder T cells. So this is we think this is important because Tregs are really important in actively suppressing immune responses. But always they must be active, or if they suppress, the responder cells are somehow modified to become the less dangerous. So what we found is that Treg really control the cell fate of responder T cells and to make the stable immunological tolerance. So this is the last slide of my talk. Now the Treg controls various the immune responses, physiological and pathological, and by targeting this population, you can either augment or suppress immune responses. And just for the treatment of autoimmune disease like rheumatoid arthritis, so what we are doing is that how we can expand Treg safely. So just controlling effector cells attacking antigen, but allowing Treg expansion by the IL-2 or other substances, you can make Treg dominant state. Or like in type 1 diabetes, if you take them out from the circulation and stimulate with self-antigen, like insulin or other antigen, and expand them, and then transfer back to the patient, now you can have this Treg dominant state. And also in the future, ideally, what we expect is that T cells causing disease, if they can be converted to Treg, that is ideal way of immune suppression. So how we can induce FOXP3 in effector cells causing disease and put them back, convert them to Treg, that is really challenging for the immunotherapy of these inflammatory diseases. So I have talked about started from a history, and so many people are involved in this project. So just recent one, the energy work was conducted by Yuka Maeda together with other people. And Shohei Hori, he is really the key player of the FOXP3 discovery in our hands, in our group. So now he has an independent group in Tokyo University. So thank you very much for your attention.
Thank you so much. While Dr. Rudensky is going to set up his computer, we'll take a few questions. So now these presentations are webcasted. So when you ask a question, look to your left. On each chair there is a microphone. And even professors may be able to do this, to lift up the microphone and turn it on when you ask a question. Because it's all, otherwise the people looking at the webcast won't hear you. So any questions? Sasha, do you want to set up? We have a question from the back there, please.
Hi, Rick Kapoor, Lund University. Thank you so much for a fantastic talk. Really, really nice overview. Amazing work. I was just wondering, what do you think are the key mechanistic differences of Tregs in their suppressive function in autoimmunity versus alloimmunity? As reports have also shown that they can do it in an alloimmune setting.
So mechanism of Treg-mediated suppression. So far, more than 20 mechanisms have been proposed, and today I talked about the IL-2 and the CTLA-4 because we are working on autoimmune diseases. So whatever it is, if it's anomaly of that molecule, if it causes autoimmunity, that is important. Very pragmatic way of the thinking. So your question of which mechanism is most important, we still believe that this CTLA-4 dependent control of APC, it's whatever inflammation, whatever immune responses, it's important. And also, but if they they further differentiate by chronic stimulation, they produce IL-10 or other immunosuppressive cytokines. So that is important for example, in mucosal immunity. But allo reaction in transplantation, still I believe that the activation of alloreactive T cells by APC, that step, that phase can be controlled by CTLA4 dependent mechanism.